welcome to my channel Omni Bio Entrance, and today we will be discussing the CUET question paper for subject Plant Biotechnology conducted in the year 2025. So coming to the first question, plants having half the somatic chromosome number than found in normal individuals are. So they have asked for half the somatic chromosome number. So they are naturally your haploids, not monoploids. So haploid will have one complete set of unpaired chromosome, and monoploid means an organism having single basic set of chromosome. Okay, single basic set of chromosome. That is monoploid. Then aneuploids means abnormal number of chromosome. It can be 2n plus 1 or 2n minus 1. Then monosomic means it is also a type of aneuploidy where one chromosome is missing from the normal diploid pair. So 2n minus 1. Then the production of haploids by anther and pollen culture was demonstrated by Maheshwari and Guha. Then plants absorb molecular nitrogen from the atmosphere, cannot absorb. Okay, molecular nitrogen, that is they cannot absorb the nitrogen gas directly. Why? Because nitrogen gas is having a triple bond, N triple bond N, triple covalent bond, and this is a covalent bond, so it is a very strong bond, and it requires high energy for dissociation of this bond. And plants, they usually lack the enzyme which are required for breaking this triple covalent bond. So they cannot utilize nitrogen directly from the atmosphere. Okay, so it has a triple bonds, making it highly stable. Then inducing the formation of vegetative organs from cells or tissues in plant tissue culture is called. So vegetative organs they have given there. So it is nothing but organogenesis. What is somatic embryogenesis means? The somatic cells of the plants, they develop into embryos in tissue culture media. De-differentiation means where a differentiated mature plant cell will revert back to form unorganized mass of a cell called as callus. That is de-differentiation. That is a differentiation of a already differentiated tissue to form unorganized mass of cell. That is called as callus. Then somatic hybridization means during interspecific cross, you isolate the protoplast from both the species. Protoplast means cells without cell wall, and you fuse them, you get a somatic hybrid. Then production of secondary metabolite you require the use of. So in plant biotechnology, for production of secondary metabolites you usually use cell suspension cultures, because this method the plant cells will be growing in a liquid medium and it is constantly kept agitated. So this agitation, it will provide uniform environment to all the plant cell, as well as oxygen availability and nutrient availability is also provided to these plant cells for their rapid growth, and as well as for extraction of this secondary metabolite. Whereas solid agar medium cannot be used. Meristem culture, it is mainly for tissue culture, micropropagation, and axillary bud is also for micropropagation. Then media that contains some chemicals with unknown chemical composition, it is called as complex media. such as if you add coconut milk or coconut water or some fruit extracts to your tissue culture medium, you don't know what is the chemical composition or how much of hormones or other nutrients are present in coconut milk or other fruit extracts. You add that to the usual tissue culture medium so that it provide hormones or vitamins and minerals that can stimulate callus or root development. So it is chemically undefined. Whereas synthetic, enrichment, selective, all these are have a definite chemical composition. Okay, how much vitamin is there, how much other potassium nitrate, calcium chloride, all these, how much amount it is there, you know in others, whereas in complex media you don't know the chemical composition. Then for obtaining pure cultures of bacteria, dash plate method is not used. Streak plate, yes, the inoculum is spread on the surface of an agar plate using an inoculation loop, and you will get individual cells. Then spread plate, that means you take a small volume of the sample containing bacteria and it is dropped onto the center of an agar plate and you spread it using an L rod. So that also will give you pure cultures of bacteria. Then pour plate means you mix the bacterial sample with the agar, that is molten agar, and it is poured into petri dish. After solidification, colonies will grow on the surface. So dip, that is not used in pure cultures of bacteria. Then select the correct statement regarding soma clonal variation. So soma clonal variation means they are variations found in tissue cultured plants. Okay, and some soma clonal variation, they are beneficial for the plant, like they provide disease resistance or improved yield. Whereas some soma clonal variation, they provide unwanted traits or deleterious traits for the plant. Now you have to select a correct statement regarding soma clonal variation. So these variations can also result in unwanted traits, yes. Soma clones have been developed and proved advantageous in several crops, of course. They have been, scientists have isolated traits for disease resistance, salt tolerance, etc. in various plants. So that's correct. Then these variations can be used to engineer novel traits. Yes. Once you get a soma clonal variant, maybe for a disease resistance, you can isolate the gene from this soma clone and you can transform another plant which is not having this disease resistance character. So it is a source of variation that can be used to engineer novel traits. That's correct. Short-term in vitro callus and cell suspension cultures result in soma clonal variation, no. But if it is a long-term, then without subculturing, or you have kept the callus in or the cell suspension culture for a very long period of time without subculturing, then the chances of mutation and variation, that is development of soma clonal variation, is more, not short term. So the answer here is A, B, C only, option 2. Then in plant tissue culture, formation of organ primordial like shoot or root in callus cell is called de-differentiation. We already saw that de-differentiation means already differentiated tissues, they will develop into unorganized mass of cell called as the callus. So now this callus, it can give rise to shoot and root. That is, the organs can be formed from this callus tissue. And this is called as redifferentiation. Okay. Now, somatic embryogenesis, I told you the development of embryos from somatic cells in plant tissue culture. Then regeneration means it is a development of the whole plant from a single cell or tissues. It is called as regeneration. Who is regarded the father of plant tissue culture is Haberland. So Gottlieb Haberland. Then the first androgenic haploid plant product by anther culture was from Datura. So first androgenic haploid plant which was produced from Datura by anther culture by two scientists, that is Guha and Maheshwari. Then the process of combining cytoplasmic genomes of one parent with the nuclear genome of another parent, it is called as hybridization. So here you are combining the cytoplasmic genome, that is your mitochondria and chloroplast, from one parent, and the nuclear genome of the other parent. So the parent which is providing the cytoplasmic genome is enucleated. That means you are not using, the nucleus is removed from this parent, and then the second parent is having the nuclear genome. So that is called as hybridization. Okay, then somatic hybridization, I told you micropropagation is nothing but tissue culture method itself for rapid multiplication of plants. Then regeneration. So somatic hybridization, what happens here is somatic cells will result in hybrid formation. So both the parents will provide the nuclear material as well as the cytoplasmic material. Both the parents will provide. That is, nuclear genome also will be there, chloroplast and mitochondria genome also will be there from both the parents. It is not like one parent providing nucleus, other is not providing nucleus. That is seen in hybridization only. Then dash molecules of ATP are required to fix one molecule of nitrogen to ammonia, that is 16 ATP molecules are required. Then symbiotic nitrogen fixing bacteria are found in. So Azolla, it is a water fern harboring the symbiotic bacterium Anabina, and Anabina is involved in nitrogen fixation. Then Netum, it doesn't show any symbiosis. Anthocirrhosis, that is bryophyte, it is having symbiotic association with the cyanobacteria nostoc. Then Cycas, it is a gymnosperm and it is having specialized roots called as the colloid roots that also have a nitrogen fixing cyanobacteria. Rixia is also a bryophyte, but there are no known reports telling that Rixia is harboring some symbiotic nitrogen fixing bacteria in it. So the answer is ACD, answer is option 1. Then important enzymes involved in nitrogen fixation are, first is nitrogenase, it's there. Peptidase is not there. Peptidase is for breaking down the peptide bonds and forming amino acids. Then hexokinase, it is involved in the first step of glycolysis, nowhere related to nitrogen fixation. Then hexokinase and dehydrogenase, not at all. Nitrogenase and hydrogenase. Yes, that is correct. Nitrogenase, it will, what is that? It is convert the atmospheric nitrogen, that is N2 gas, into ammonia, which is used by living organism. And hydrogenase so is also involved in nitrogen fixation, because during nitrogen fixation nitrogenase produces hydrogen gas as a byproduct. So this hydrogenase, and as a result of this hydrogen gas production, there is energy loss for the cell. So hydrogenase will help in recovering the energy lost, and it will recycle the hydrogen. Then next one, pomato. A hybrid of potato and tomato was produced through nothing but somatic hybridization. Shoot tip culture means the shoot tip is cut off from the plant, and it is inoculated into suitable medium under aseptic conditions to regenerate into whole plant or to form callus. Then anther culture is mainly for the production of haploid plants. Okay, so the answer is somatic hybridization. And this potato and tomato, they are incompatible, sexually incompatible species. So this technique, somatic hybridization, is usually used to create hybrids from these plants. Then arrange the following step in the process of nitrogen cycle in the correct sequence. So first is nitrogen fixation. So nitrogen from the atmosphere is fixed by nitrogen fixing bacteria to ammonia. Okay, and through lightening also nitrogen fixation is happening. So that is the first step. Then comes nitrification. So nitrification means ammonia getting converted to nitrates. That is nitrification. NO3 minus. Okay, ammonia getting converted to, okay, first to nitrites, that is NO2 minus, and then to nitrates, NO3 minus. And this NO3 minus, it will be easily absorbed by the plant, that is assimilation. So after nitrification, you have got assimilation. Then comes after assimilation by the plants, the herbivorous animals, they will eat the plants, and nitrogen is getting accumulated in the animals also. And when these animals excrete waste or when they die out, the decomposers will convert the nitrogen present in the animal excrete or the dead body of animals and plant by the process of ammonification. They will convert these nitrogen compounds into ammonia. Okay, so first nitrogen fixation, then nitrification, then assimilation, then ammonification, and then finally denitrification, that is by denitrifying bacteria, where the nitrates are converted to nitrogen of the atmosphere, and thereby the nitrogen cycle is completed. So this cycle you have to remember for the rest of the paper, because so many questions they have asked this directly as well as directly regarding nitrogen fixation. So first will be nitrogen fixation, then nitrification, then assimilation, then ammonification, then denitrification. Okay. So that makes A, B, D, E, C. A, B, D, E, C is option 3 is the answer. Then virus-free plants can be grown by apical meristem culture. Why? Because apical meristems, they divide rapidly, even faster than the virus. So the virus will not be able to replicate and spread in them. Then they have low lack of vascular connection. Some phloem tissues may not be well developed in apical meristem cells. So, and most of the plant viruses, they spread through phloem. So lack of proper vascular connection means the virus cannot easily spread to other places via apical meristem. So to produce virus free plant, you can use apical meristems. Then next one is match the following somatic hybridization. So somatic hybridization is a fusion of protoplast from somatic cells. So A2, that is there in option 1 and 2, so 3 and 4 are wrong. Then parthenocarpy means without fertilization you are producing fruits. So seedless fruit without fertilization, so B3. Okay, so 2 is the answer. Then micropropagation means multiplying multiplication of plants without sexual reproduction, so C4. Then single cell production, you require cell suspension culture. So cell suspension, why you require that means cell suspension culture, it is produced by growing single plant cells or using friable callus, so that they in suspension culture upon agitation they will be easily separated. Okay, and there will not be aggregation of the callus also if you are using friable callus tissue. So single cell production, you use cell suspension culture, so D1. Then next one, arrange the following steps for plant tissue culture in correct sequence. Okay. So selection of desired material and suitable nutrient media, yes, that you have to do. You have to decide which material you are going to use as explant, whether it is apical meristem or shoot tip or leaf pieces, etc. You have to decide. Then you have to choose which nutrient media you will be using, whether you will be going for the MS medium or the Gamborg's medium or white medium or whatever. So that you have done, then you will prepare the media and keep it aside. Then next will be, you have to bring the explant from the outside grown plant. So you have to surface sterilize the explant, because the explant bought from the outside will have microorganisms on it, so they can contaminate the culture medium. So to prevent that, you will surface sterilize the explant. Then after that, under the laminar airflow hood or under aseptic condition, you will inoculate the explant into the suitable media. Then the next is transferring of growing cultures, that is nothing but subculturing. So as the plant grows, the nutrient media will become exhausted. So you have to transfer the plant to a fresh medium. Okay. So transfer of growing cultures. Then a transfer of plantlets to soil in pots. Then the next will be the acclimatization, that is the plantlets which have developed the root and the shoot, they will undergo the hardening process, so that they, or acclimatization, so that they can be moved to the natural soil in pot. So the first will be A, C, B, A, C, B, D, E, that is option 2 I think is the answer. Okay. A, C, B, D, E. In the next one, in nitrogen cycle, ammonification is the process of generating ammonia from. So ammonification I told you by decomposers. So what they do is they will convert the amino acids, that is they will come the amino acids will have these amino groups that having the nitrogen, so they will convert, or else they can convert the proteins or nucleic acids which also harbor nitrogen to ammonia. Okay, that is ammonification. So it is generating ammonia from amino acids. It is done by the decomposers, that is bacteria, fungi, etc. Then match the following. Gene inhibition means you inhibit the faulty gene, or make the gene in such a way that it will not produce any product because it is a faulty gene. So that is gene inhibition, that you can see in disarm the product of faulty gene. So it can be like blocking or reducing the expression of one specific faulty gene, so that the harmful product is not produced. So A2 means option 2 itself is the answer. Then gene editing is by CRISPR-Cas technology. It is because it is very precise and it will edit your gene according to whatever you want. So B will be 3, and gene targeting. So here you target one gene, and because it is not, just because it is non-functional, you are targeting that gene and you are replacing it with a functional gene. That is gene targeting. So that will be replacement of non-functional gene with a normal gene. So C will be 4. Then gene augmentation. Augmentation means adding something good. Okay, so D will be 1. That is addition. See here, adding. Addition of functional gene to their genome to replace missing product. So D will be 1. Then next one, conversion of ammonia to nitrates. That is called as nitrification. Answer is option 3. I told you from nitrogen cycle they will keep on asking these questions. So nitrification means where the soil bacteria, they will convert this ammonia to a form that is easily absorbable by the plants. That is called as nitrification. Ammonification, I already told you, by decomposers they will convert organic nitrogen back to ammonia. Assimilation means the bacteria absorb the nitrates, and no, not bacteria, plant absorb the nitrates. That is assimilation. And denitrification means the process where the nitrates are converted to gaseous nitrogen and returned to atmosphere. Then full form of CRISPR, a term used in gene editing, is clustered regularly interspaced short palindromic repeats. That is the full form of CRISPR. Then choose the correct statement regarding plant tissue culture. So organogenesis is inducing the formation of various vegetative organs from cells or tissues. Yes. Vegetative organs means like shoot and root from the cultured explant. Formation of root and shoot from cells and tissues means those cells or tissues that you are putting in the tissue culture medium. That is organogenesis, that is correct. Formation of mass of undifferentiated cells from callus is redifferentiation. It is not correct. It is de-differentiation. We have been already discussing differentiation, de-differentiation, redifferentiation till now. So that is this is also a continuation of that question. Then cytoplasmic hybrids are prepared by taking the nucleus from one parent and cytoplasm from both the parents. Yes. So hybrids, they are produced by fusing protoplast containing the nucleus of one parent but cytoplasm, that is mitochondria and chloroplast, from both the parents. So that is correct. Then relative concentration of growth hormone play important role in organogenesis. Yes, of course. If you have a high cytokinin to auxin ratio, then it promotes shoot formation. Or if it is high auxin to cytokinin ratio, then roots will be developed. So concentration of growth hormones is playing role in shoot and root formation. So that is in organogenesis. So A, C, D is the answer. So option 4 is the answer. Then choose the correct statement regarding homozygous diploid plant. Okay. Homozygous diploid, they are usually developed from haploid plants. Okay. Now read the statement. They are produced by doubling the chromosome number of haploids. Yes, haploid is having only one set of chromosome. You add colchicine to it. Colchicine will cause a doubling of the chromosome. So you will get homozygous plants. So they are produced by doubling the chromosome number of haploids. That is correct. Doubling of the chromosome enable the recessive trait to be expressed. Yes. Now, if you are seeing a standard diploid plant, it is having a recessive trait, that is small a, which will be masked by the capital A, which is dominant. Now, a haploid plant, it can carry either capital A or small a. Now, here they have told recessive trait. So, we will see this recessive trait. So the haploid plant will have recessive trait. Now what you do after haploid, you will double this chromosome by adding colchicine. So when the chromosome is doubled, it becomes capital A. It will not become capital A, it will remain small a itself. This only one set of chromosome is there in the haploid. This is doubled means you will get small a only. Okay, so small a means homozygous recessive character is expressed. So B is correct. Chromosome doubling is done by ozone treatment. No, ozone doesn't play any role in chromosome doubling. Colchicine is used, so D is correct. It will inhibit spindle formation during mitosis. So the chromosomes will not be able to separate to different cells during the anaphase, and this will result in chromosome doubling. So what are the answers? A, B, D, A, B, D, 1. Answer is 1. Then next one, transgenic plants developed by introducing Bt gene in crops like brinjal, maize and cotton provide resistance to insect pest. Okay, these Bt gene, it will produce the cry protein. Cry protein are toxic to insect. When the insect eat these plant tissues having the cry protein, they will inject the toxin also inside them, and when the cry protein reaches the insect gut, it will get activated by the alkaline nature in the gut, and it will create pores in the intestine, and it will lead to the death of the insect. So that is the answer. So it is for insect pest. Then which of the following converts again nitrites to nitrates? Okay, nitrites to nitrates. So first is what? Ammonia. Ammonia is formed. Ammonia is converted to nitrites, NO2 minus. So that is by nitrosomonas and nitrococcus. So that is how nitrites are formed. They have asked what will convert nitrites to nitrates. So nitrites will be converted to nitrates, that is NO3 minus by nitrobacter. Okay. And Clostridium, it is a denitrification bacteria. So that is also not the answer. So nitrosomonas, nitrococcus, they will convert ammonia to nitrites, nitrobacter, nitrites to nitrates. Then San Noem first successfully cultured gynogenic haploid plants from unfertilized ovaries of, he used barley for gynogenic haploid plant production. This is an like gynogenic means alternative to the pollen androgenesis, pollen culture for the production of haploid plants. Then nitrogen fixation occurs with the help of symbiotic bacteria. So nitrogen fixation with the symbiotic bacteria is seen in your leguminous plants, that is members of the Fabaceae plant. So pea is a legume, lettuce is not, beans is a legume, tomato is not, black gram is also a legume. So A, C, E is the answer. So 4 is the answer. Then in biological nitrogen fixation, conversion of dinitrogen molecule into ammonia is carried out by the nitrogenase enzyme. Okay, we are discussing that from very beginning, so you know it is nitrogenase. So I am not going into again and again, I am not repeating it. Then choose the correct statement regarding cytology of haploids. So a haploid in Arabidopsis will have 5 chromosomes. Of course, the 2n number or diploid number of haploid is 10. So the haploid will have 5 chromosomes. That's correct. Then haploids are found as bivalents. How can that be? Bivalent means chromosome from my chromosome diagram is very bad, but still I am okay. I will try. See here, bivalent means this is your paternal chromosome and this is your maternal chromosome. They come together during the zygotine stage of prophase 1 of meiosis to form bivalents. In haploids, will you ever find bivalent? It has only one set of chromosomes, so only one will be there. Where is the other pair? Where is the pair from the maternal or paternal plant providing the homologous chromosome to form bivalent? No, it will not be found. So haploids are found as bivalent at metaphase 1 of meiosis is not correct. So haploids are found as univalent at metaphase 1 of meiosis, that is correct. Then the haploids in maize will have 10 chromosome. Yes, when maize is having diploid number of 20, the haploid number will be 10. The haploid in maize will have 20, no it is 10 chromosome. So E is also not correct. So A, C, D will be the answer, that is 3.